Gene expression is really important because it kind of changes how genetic engineering actually works. Because most of us think that when we do genetic engineering, we'll insert a gene and then we're done because it will be coded for. But sometimes the gene expression of the actual gene is it's not expressed properly. Sometimes the gene won't even be coded for, and sometimes it'll act like it wasn't even there. So this this kind of gene expression messes with how genetic engineering actually works, and it messes with the efficiency. And today we're going to try to see why there's actual genetic expression. There's a difference, even though the gene is still there. Now we have two parts to the gene, and the main part that we're going to be talking about is the transcribed region, because that's the part that's actually coded into. The regulatory region is still very important because it allows the transcription factor to bind and tell the polymerase to actually code. So here, if we were to have a positive transcription factor, we put a polymerase on, and then it would unwind the DNA, read it, and then wind it back. And then we have messenger RNA. This carries information of this DNA segment. And then we would have a ribosome here. And it would actually read and then produce a protein out of that information. So here we're going to have that type of protein that is created from this specific region of the DNA. And we also have mRNA destroyers that get rid of this mRNA because the mRNA is still there. Once it's read, it's not destroyed. And it can be used over and over again. And the mRNA destroyer limits the production of proteins. And we also have negative transcription factors, and they do the opposite of positive transcription factor, and they also inhibit the positive transcription factor from actually binding because they take the space. And here you can see it just stops the polymerase from actually transcribing. And then in the next, if we were to do the next gene, it's very similar. But this time you have two transcription factors, and you need both of them in order for the polymerase to actually code. So here, I'll show an example, and then here codes. But if we were to have a negative transcription factor, neither one of these could actually bind. And since we have a new mRNA, there's going to be a new protein that's created. So here we have a hexagonal looking protein. And I don't think we really need to go more in depth about this because it's straightforward. So here we're going to actually have a simulation of how it would actually work. You would have random floating polymerases, and you'd have positive transcription factors, and they'd bind, and they randomly find their way. They aren't like seeking their own path. They don't find there isn't like a path to go. They just randomly float around until they bind. And the affinity changes how well they actually bind. Sometimes, for example, if we put the affinity to medium, the positive transcription factor would stay on, but sometimes it gets kicked off because the affinity is not high. And the same can go with polymerase. If we were put it at low, it would bind, but it would be impossible for it to code. And if we were to add a negative transcription factor, now you can kind of see how gene expression is modified, because if we were to have a medium concentration of positive transcription factors but a very high negative transcription factor, then even though they have the same affinity, then it's more likely for a negative transcription factor to actually bind, and that would just stop the gene from actually being expressed fully. And sometimes it wouldn't even allow the gene to be expressed if the affinity is too high and the positive transcription factor's affinity is too low. So if we were to do this in a cell, here we have bacteria cells that have a gene that codes a protein that's fluorescent green. Now, there are many different ways to alter how the gene is expressed. Keep in mind, they all have the same gene, but they don't have the same amounts of positive transcription factors. Some have a bit more than others, and there's also that part of chance about how affinity actually changes how they react to DNA. So if we were up the transcription factor, that'd make it so that the positive transcription factor would actually just stay on to the DNA segment, and it would not come off as easily. But if we put it to high, see it go up a bit, and then the mRNA destroyers gets rid of the mRNA, which we need to code for proteins. And the more mRNA is, the more mRNA there are, then that means it's more likely that it will be coded by a ribosome. So if we were to lower that, that would also increase the average protein level. If we're also to increase the positive transcription factor and the polymerase affinities with the actual DNA, that would also lead to an increase in protein because they would be more efficient and they spend more of their time actually coding instead of trying to find their way to the DNA. Up that, a little spike, up that, go up a bit more. And then degradation is also a very big thing with proteins because the protein won't all be there forever. It'll degrade, and how fast it actually degrades will limit the gene expression. So if we were to put it at slow, then there'd be you can see here it's the average protein level is huge because the proteins stay there more often. And now all of them are fluorescent green. But keep in mind that these genes are the same, but we're only changing these three types of factors: the degradation, the affinities, and the concentration. If we were to make it so that it was more in favor of silencing the gene, so we would make the positive transcription factor lower, more mRNA destroyers, lower the positive transcription factor and the polymerase affinities, then you can see quickly that without even having to put the degradation to fast, that the average protein level is just dropped. And if you put it fast, it's like putting salt in the wound. There's really no point. You can see here that they all have the same gene, but these three factors are also very important for gene expression. And in real cells, you can see this. With E. coli, they had a green fluorescent gene that coded for green fluorescent protein. And although they are all genetically identical, they don't all have the same amount of gene expression. Some of them are very bright green. This one's very dim. These are dim. These are bright green. So they might all have the same genetics, but they won't be expressed the same. And this shows that genetic engineering doesn't only hinder on genomics, but also transcriptomics, because the way that the genes are actually transcribed and they're actually read can change the gene expression. And sometimes a gene is implemented, but it won't actually be transcribed properly because of negative transcription factors that limit how much is actually coded from the gene. And sometimes we are not able to actually find the true purpose of a gene or what the gene actually does because of these transcription factors. It introduces noise when we're trying to find out what the gene actually does. So to wrap it all up, transcriptomics and genomics are big parts of genetic engineering, and how a gene is actually read and transcribed really changes how genetic engineering functions as a science and how much it relies on transcriptomics as another science in order for genetic engineering to actually grow and actually be effective. We need to learn transcriptomics before genetic engineering.